All right, so I think you can get started now, and anybody who comes in late will hopefully we can catch them up if they have questions at the end. So welcome to everybody. My name is Abigail Sullivan, and I'd like to welcome you to this session where Dr. Ranjan Krishnan is going to be presenting his work on the spread of starry stonewort and the role of climate conditions. So Ranjan is currently the Invasive Species Ecologist Fellow with Indiana University's Environmental Resilience Institute. And he was formerly a postdoc affiliated with the University of Minnesota Twin Cities, where he studied the spread dynamics and impacts of aquatic and terrestrial invasive species, including starry stonewort. So today, Ranjan's going to be sharing with you some brand new data that was just collected in August. So as it did for many things, COVID altered the original fieldwork plans for this project this summer, but we were able to fit in an abbreviated data collection effort, and Ranjan has some really interesting data to share with you today. So please feel free to type in your questions into the chat box as you think of them throughout the talk. We'll stop at about 12:45 to begin the question and answer section. So I will let Ranjan take it from here.
Thank you a bunch, and FYI, since I don't think Abigail introduced herself, this is Abigail Sullivan. She's also a co-PI on this project. She's helps out on a lot of parts of it, and is actually on my very first slide. Oh, let's see if I can get this to work. And I want to say that I'm excited to talk to everybody about this work, and I'm going to talk about just a component of this project, but it's actually a much larger project that thinks about the spread and impacts of starry stonewort, and not just on the ecological side, but we're really interested in other components like the impacts on ecosystem services and other values associated with these lakes, and even the management and decision-making processes around starry stonewort. And so Abigail and a number of other collaborators have been really important in those components of the project and the project overall. Also I'll say it's a collaborative project between both MACERC in Minnesota and the Indiana University Prepared for Environmental Change Grand Challenge down here in Indiana. And I just want to start by saying thank you to all those other people who've been really critical to the project. And so today I'm going to start with just a quick outline of what I want to talk about today. There'll be a little bit of an overview of starry stonewort and its background, although I imagine many of you are already somewhat familiar with it. Then I want to give you a little bit of the background and rationale that were involved in sort of the conception of the project. And then the actual efforts that we took, the approach that we have. And then, as Abigail said, we'll give you some of our new initial results, really just came out in the last month or so, which we're really excited about. I think that it's been an interesting project, and I think there's been some interesting results from it. So to start off, starry stonewort, or also known as Nidalopsis obtusa, that's its scientific name. It's a green macroalga. It's an algae, but it has this sort of complex three-dimensional structure, much like vascular plants. And if you went out and you saw it in a lake, it would look like you might imagine a plant to look. Starry stonewort, it's also, it's a charophyte. It's closely related to the stoneworts or musk grasses, many of which are native to Minnesota. In particular, the caras are species that many people might be familiar with. And these types of plants are often pretty ecologically important, both because they can really influence water quality. Sometimes they can even be considered a sort of an indicator of water quality, of usually high water quality. And they're also important habitat for other species like invertebrates or fish. So if you have shifts in those communities, you're likely to have other ecologically important impacts as well. So starry stonewort, it's a relatively new invader, both to North America. It was first identified in 1978. And then even more recently, it was identified in Minnesota only in 2015, though it's quite likely it had been in Minnesota for at least some period of time before that. It's also expanding quite quickly, both in terms of spreading across the United States and entering into new lakes and spreading in that sense, but also in individual lakes. There's been quite a lot of spread in recent years as people have been watching it. I know Wes Glisson gave a really nice talk a couple days ago, really focusing on that sort of within lake spread pattern and showing a lot of nice data, really getting at that question. And because of this rapid spread, starry stonewort has been considered an important AAS management concern in much of the Midwest. If you want to look, it's probably a couple of years old now, but this is sort of a general distribution of starry stonewort across the United States and Canada and sort of these upper northeastern and midwestern states, mostly or generally in the Great Lakes region. But there have certainly been at least a handful of new additional locations that it's been identified since this map came out. And in Minnesota specifically, I believe we are now up to 16 separate lakes that have starry stonewort, and a hat tip to, I believe, about 4 of those lakes were actually identified via Starry Trek, a really cool MACERC supported outreach and citizen science project. If you don't already know about that, you should check it out. You can see the website there, or just Google Starry Trek. I'm sure you'll find it. And talk to anybody at MACERC. I'm sure they'll have information for you as well. And so when we think about starry stonewort and we tend to think about its impacts, they largely are driven by the fact that starry stonewort can grow in these really dense accumulations. They can get extensive, they can get really thick, in particular, they can get really tall and often come really close to or even over the top of the surface of the lake. I know, so in particular, a lot of those related species like the caras are often only a few inches or a foot or two in height, but then starry stonewort can grow much taller. I've certainly been in patches that are 10 feet deep or 10 feet thick or tall, however you want to describe that. And that, that really large, really dense amount of biomass can cause issues like it's really easy for boat props to get caught up in it. It's hard for boats to move through it. You probably don't necessarily want to go swimming or fishing in areas that are highly, highly filled with starry stonewort. So that high abundance tends to have these impacts on sort of recreational usage of starry stonewort. The other major impact that I tend to think about with starry stonewort is its impact on native communities, or native plant, well, both plant and animal communities. When you tend to have a lot of starry stonewort, it ends up often being in monospecific or almost entirely starry stonewort communities. They're not these sort of like complex mixed communities that you likely have previously. And when you sort of have this reduction in diversity, you also tend to have reduced resilience of those ecosystems to other stresses that might arise, and a reduction in sort of attendant ecosystem services. So those are the general impacts that we've seen or that we tend to think about with starry stonewort. And so as I was saying, or as I mentioned, I've worked on this species for a little while, and a few years ago, I was really interested in trying to think about how can we monitor the spread and expansion of starry stonewort. And so I established some monitoring transects, particularly in Lake Coronis in Minnesota. And the idea was we essentially went to an area that we could see both starry stonewort adjacent to native plant communities. And then we essentially just placed fixed transects, or, at each end of this 30-meter long line, we placed a fixed, essentially a fence post that we could then run a long tape measure or transect tape between them. And then we can, we can swim along that transect, and we can ask sort of at every meter or every other meter, well, what is the cover of different plants there? Particularly starry stonewort, but also, any of the native plants, how much is there? And, that way we can see, is it dominant by a starry stonewort or native species? So we started this at Lake Coronis, and the way we did this is we actually ran 3 parallel transects right next to each other, about 1.5 meters apart from each other. And we set up sets of 3 in 5 locations across the lake. And that's essentially what you see in all of these, in this plot here. And you can see each of those dots represent a single square meter and the percentage cover of Nidalopsis obtusa or starry stonewort. And you can see what we did is we always started them in a starry stonewort dominated habitat. And then we ran the transect into an area that was still dominated by native plants. And then we tried to set it up so that there was a point with a sharp transition that kind of indicated the invasion front, sort of the leading edge of the invasion, because that would be the easiest place to measure sort of the spread forward over time. And so for any of you that were perhaps at the showcase last year and have been following starry stonewort, you might have seen some of this data that was presented by Carly Wagner from these Lake Cronus transects, where it was actually, she showed this really dramatic spread over just 2 years. These transects went from being mostly native species to almost entirely dominated just completely by starry stonewort. It was this really dramatic rise. And that was a really important result for us to see, and to see that that spread could happen incredibly quickly. And this, mostly fits with this idea that this sort of dogma we have about invasive species, where, initially they're absent, and then they're just probably really quite rare, and there's sort of a lag before they can start get going. But then once they sort of establish well, the spread is really rapid and really complete. And then once you have, expansion of starry stonewort or of any invasive species, it then is there and it's complete and it's sort of there to stay. And that's, the sort of idea that I think a lot of people are working with. But I'd spent enough time at particularly Lake Coronis, but at other lakes as well, to see that, it wasn't always that, it wasn't always that way. It wasn't always this like super, super abundant, over the top of the surface, really gunky places. Like, I'd go to the same locations in subsequent years where, previously there'd been super high abundances, but then in the next year, there'd still be starry stonewort. It'd still often be, pretty high coverage, but it would just not be quite so abundant. It wouldn't come all the way to the surface. And mostly it wasn't, in my opinion, quite so much of a nuisance. It'd be a few feet below the surface. You could still drive your boats around. And so I was interested, like how consistent is the abundances of starry stonewort? Does biomass ever go down? Does it ever, when does it go up? When does it go down? Is that predictable? Can we figure out, when we're gonna have more or less? And in particular, are there things like climate conditions, like, winter severity or growing season length that might actually help us understand and predict when we're gonna have really major starry stonewort years or kind of lesser starry stonewort years. And so that was all, that sort of impression that I had from just being out in these systems was kind of the impetus for, wanting to look into these questions. And so, so I'm going to talk about our approach in 2 components: the hypotheses that we want to think about, and then the activities, the actual experiments we've been running. And so for hypotheses, particularly with relationship to climate, we're interested in, well, is there an effect and what would it be? And so could we predict things like longer growing seasons might allow starry stonewort to expand? They're really good growers. And if they have more time, more light, it's warmer, that'll help them expand and just really take over. Alternatively, what we've seen with a number of other invasive plants, particularly curly-leaf pondweed, is that they are really successful because they are good at tolerating sort of colder, early season, darker conditions. And because they can tolerate that, they have this sort of leg up, this head start over the native species. And so we could predict or hypothesize that something like a cold spring is going to provide this advantage to invaders. And so you'd expect more starry stonewort in years that are a little bit colder. Conversely, it could go the opposite might also be true then, that if conditions are warmer, that would actually erode some of that head start that the invaders have and make native species a little bit more competitive, a little bit more resilient. to the invasion. And so we wanted to test out these types of hypotheses, to try and relate spread rates, biomass, and climate conditions. So the way we did that is by establishing more of these monitoring transects, just like I sort of described a moment ago, but now we're going to do them across a much broader range of lakes and across a latitudinal gradient that goes all the way from Indiana up to the northern parts of Minnesota near Bemidji. And that actually reflects most of the latitudinal range that you see of starry stonewort in North America or in the United States. And so once we set up those transects, we would be able to measure plant cover, both of starry stonewort and native species. And then by going back and measuring in repeated years, we could also measure the rates and amounts of expansion of starry stonewort. And same, for native species. And then alongside those sort of plant cover measurements that we do on scuba, we also go out and essentially with a rake, we can pull up biomass itself and actually measure its weight to get a sense of both the sort of seasonal patterns of when is it more or less abundant, and then annual patterns of, are there good years and bad years for starry stonewort. So that was our general approach. So as I mentioned, I'd started this work a little bit before, or when I was still up in Minnesota. And so we had a little bit of pilot data from a couple lakes, Coronis and Moose, where in particular, we were measuring the biomass. And you could see a couple things in this figure. One is this like nice seasonal pattern. There's, kind of low biomass in the spring, peak biomass in sort of August and September, and then a winter die-off. So there's clear seasonal patterns. But then if you look across the different lakes, Moose is in green and Cronus is red and black. You have lake-to-lake differences, and even within Coronis across 2 different years, that biomass can be different. Again, showing that there is some heterogeneity, some variability year to year. We don't know necessarily what the conditions were in any of these given years, but it was just showing that there is some heterogeneity for sure. And so our plan, again, as Abigail mentioned, was to go out and do a big field campaign this year and essentially collect the data to draw this kind of a figure for all 9 of those lakes that we wanted in our gradient. But unfortunately, COVID happened, and so we were unable to do that major field campaign. But we were able to get out and do a really, really constrained, very quick, just couple-week field set of field surveys, and we went to each lake and were able to get their biomass at sort of peak biomass in sort of that middle of August. And here what you can see is we're just plotting all of our 8 or 9 separate lakes in that sort of peak biomass period. And again, what I really want you to take away from this is just there's a bunch of variability both across lakes, and the difference, so different lakes are in different colors, different years are different dots, and point is there's just a fair amount of heterogeneity. And then I've also circled all of the 2020 data points, which sort of suggests that 2020 was a relatively lower biomass year for a lot of our lakes, with the single exception of that top point, which is actually Lake Coronis. But for a lot of locations, it was a relatively low biomass year. That same data, you can plot it a little differently. So for each lake, now we have 2019 and 2020 data. And again, you could see a number of lakes where it's going down, although there's a few where it's kind of a wash between years, and then one lake where maybe you actually saw a little bit of an increase. But again, heterogeneity is the main story that I want you to take away from this. And again, it was a relatively lower year in 2020. So that was biomass in areas that had already been dominated by starry stonewort. We picked locations with starry stonewort and checked their biomass there. So the question is, does this influence or can we correlate that with the expansion patterns of starry stonewort? So here we're back at Lake Coronis, that same plot that I showed you guys a couple of moments ago with that dramatic shift from 2017 to 2019. And now if you add in data from 2020, you can see there's still quite a lot of starry stonewort, but it's actually a bit more heterogeneous than it was in 2019. And there were a couple of locations that were really just completely dominated by starry stonewort in 2019. But you can actually see a recovery or a resurgence or whatever you want to call it. We're actually seeing increase or decreases in abundance of starry stonewort, which are also largely aligned with increases in native species cover. So that was really interesting that, while we'd seen previously just this huge expansion in starry stonewort in Lake Coronis, we're now seeing in 2020, which is maybe like a less good starry stonewort year, you're actually seeing these reductions and potential increases of native species. If we then go to our full transect or our full set of lakes from all the way from Lake Syracuse is our most southern lake in Indiana to Moose, our most northern lake in Minnesota. In 2019, when we started this, you can see again, there are those nice sharp boundaries going from starry dominated to native dominated. And then if you add the 2020 data, you can see there's actually still quite a lot of starry stonewort, or sorry, a lot of native species on a lot of these transects. And I totally get that this is a really hard set of images to look at, don't worry. What I've done is I've essentially subtracted the 2019 data from the 2020. So you can just look at how these transects have changed. And so what's important here, let's see, these areas where you see orange and yellow colors, that's where starry stonewort increased, where you kind of had expansion of the invader. But then what's really interesting is these other areas where you actually have these bluish colored areas where you actually have reductions in starry stonewort, and you have you have less than there was in previous years. Green is essentially staying the same. And then what's also interesting, this is, the changes in the amount of starry stonewort, but now I can flip it and I can look at the changes of native species. And here we actually can see there's a lot of oranges and yellows, and you're seeing expansion of native species on a lot of these transects, even in areas where there were previously the invaders. So that's kind of going against this idea that, the invaders, they come in, they take over, and then they just expand, and it's just a constant progression of more invasion. I think what's interesting here is it's a lot more heterogeneous than we would have expected. So then we wanted to try and integrate those two pieces of data, those two components, and think about, is how good of a year it is, like how much, if we look at the starry stonewort biomass in like a heavily starry area, and that's my measure of how good of a year it is that also predictive of, the spread amounts, like how much is it expanding across the whole transect and into the native dominated areas. And here you can see, on the x-axis is sort of starry stonewort biomass, which is my measure of sort of how good of a year is it for starry stonewort. And then on the x-axis or the y-axis vertically is sort of the change in total cover. And you can see when it's a really high biomass year or biomass location, you're getting really strong expansion. But then there are quite a few number of locations where starry stonewort wasn't growing very well, and that's exactly where you're seeing reductions in cover across the whole transect. So those are just patterns that we're seeing at the moment. We don't necessarily, that doesn't necessarily explain like, well, why is it better in some locations and expansion is more in some locations and it's reducing in other locations. And that's really where we want to start going, trying to think about those questions of explaining those patterns, being able to predict those patterns. And in particular, we're interested in this idea of climate as a predictor, those ideas of, winter severity, summer mildness. And so to get at this question, what we've done is we just went to NOAA and looked at sort of from their climate data set, we tried to get historical temperature data, and we just essentially averaged out, the winter high temperatures and summer high temperatures for each specific year when we're sampling. And then we tried to correlate that with how much starry stonewort there was at that location. And again, we can do that across each of the years that we have starry stonewort biomass data. And it tends to look like this, which is largely very messy. You can see a lot of lake-to-lake differences. You can see a lot of year-to-year differences. And you can see a very weak pattern that, with increasing temperatures, you're actually getting slightly less starry stonewort. But again, I want to really emphasize it's a really weak pattern. There's a lot of things that we haven't had the time to sort of consider in this analysis, such as how much starry stonewort was there in the beginning? Was it already dominated, or is it a pretty new invasion? What does the native community look like in these locations? Are there other water quality issues that need to be considered? But I think it's important to recognize, this is the kind of analysis we can do. We can, now that we have sort of biomass and expansion data, and we're going to try and get some other water quality data, we can start aligning all that with climate data, and particularly as we get more years of data, that's going to offer an additional level of variability along with our latitudinal gradient to help us think about these sort of climate influences. And that's what I'm really interested in getting to in the next couple of years. So that's what I wanted to present. Just a couple overall conclusions, what you should really take away from this mostly, and more than anything, is that there is really high variability in biomass between both years and lakes, and that largely correlates with the spread rates, the expansion of starry stonewort, which is also variable and inconsistent. In fact, some years you actually get reductions. Although, when you have sort of better starry stonewort growth, that's when you see more expansion. Perhaps that's not surprising, but it's interesting mostly to see that there's also bad years. I think it's pretty likely still that climate is going to influence starry stonewort growth, but it still remains a little bit to be seen how much, what that effect looks like, what particular climate metrics or components influence it. Is it going to be positive or negative? I think there's going to be an influence, but we're still working on trying to understand those questions. And then a couple other sort of big questions that I think this leaves open and we're posed or poised to try and think about is, we saw those native species also increase in 2020. So is there a role for climate in sort of the competition between native species and invasive species, and how might that play out? And then, from there, what are the potential impacts of climate change? If the climate shifts in Minnesota, is that going to make starry stonewort and potentially other invaders worse or potentially better? So I think we need to understand that initial climate relationship, then we can start linking it to the climate modeling data to really get at these sort of future prediction questions. So that's everything I have to say. Again, I just want to say thanks to all of the collaborators, partners, funders. Particularly, I want to shout out to Carolyn Kalinowski, who has really been the lead on all of this field work, and without whom none of this stuff could get done. Everybody else also super important, but for this field work, Carolyn is the person. And also, I'll just say thanks to all of you for giving me your time. And if you have any questions, we have, I think, a good chunk of time that we can hang out and talk about whatever you want to talk about.
So there are a few questions, a couple of questions. If anybody has questions, I encourage you to take a couple minutes, think about them, and write them in the Q&A box, and I will read them to Ranjan. But just to start out here, so there was one question from Alicia about how the experimental latitudinal range compares to its native range. And I'm guessing that this is, and Alicia, if this is wrong, you can feel free to type and correct this. I'm guessing you are asking how this range compares to starry stonewort's native range, and I'll let Ranjan address that.
Gosh, I honestly, I'm trying to just imagine the 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 maps in my head, and I'm having a little bit of trouble. Mostly because I know that the two sort of major chunks of its native range are um, sort of Western Central Europe and then also Japan, and I can't for the life of me think about how those align latitudinally. Um, and uh, I guess I'm at a bit of a loss. I'm not honestly sure, but it seems like a question I should know the answer to, and I will definitely look it up. But yeah, honestly, I can't venture a guess. I don't know, Dan, if you're on this and you want to like have an answer and type it into the chat box, I'm sure Abigail can relay that. But I like to believe that we have a pretty decent latitudinal gradient, but I honestly just don't know.
So second question here is related to treatment. So this is in the context of a specific lake. So somebody's asked, was chemical treatment used on initial management, and did it have a negative impact on natives that opened the area faster to expansion of Starry? And they've said that on Grand Lake, they've not used chemicals, and Starry is still highly mixed with natives and is not expanded outside of the initial access site. So I can repeat that if you need.
Well, I think I'm getting the gist of the question. You know, there's this idea that if you do some sort of management treatment, particularly ones that are non-specific, that they impact both native species and invasive species, it could it'll kill the invader and sort of give you the short-term benefit, but in the long term, it might actually work the other way because you've now also gotten rid of all these native species that would have, been able to compete and provide some resilience to continued expansion. And I'm going to say with most of the locations that we are working in, we tried to bias towards locations that have had minimal to no uh, management, particularly chemical management. We don't know that 100% for a fact, but in most of these lakes, we would often see other areas that had been treated, and that's that certainly influenced our decision about where we put transects. And so mostly, I think that these are kind of honest, results in that they weren't necessarily highly impacted by management, but with a couple exceptions. Little Mosquito Lake, which is in Wisconsin, sort of near Milwaukee. We do feel pretty confident in that lake. They had a treatment, but I don't think it was chemical. I'm pretty sure it was a mechanical harvester. And a number of our locations we think had been harvested, and those were some locations that we actually saw real reductions in starry stonewort and resurgence of native species. So at least in that specific context, I think it was getting rid of the starry stonewort actually ended up, the native species were able to recover. I don't know if that would happen in all cases, and I am less confident. I'm actually honestly surprised that it worked that way with the mechanical harvesting. If I might have bet the other direction, honestly. And I don't know if there would have been a difference to if they'd been using chemical treatments. Chemical treatments, there's this idea that they can be specific to algae and not vascular plants. And so, that specificity, which a mechanical harvester wouldn't have, might offer a different way, or might work out a little better as well. So I guess a long-winded answer to say, like, I think that this is a real concern. I don't think that that's like necessarily what we were seeing in any of these plots, again, because I think there was relatively minimal management happening in our plots or our transects.
So just so that everybody knows, circling back to that previous question about the latitudinal range, Wes has typed in that they're fairly similar. So just so everybody knows.
Excellent. Thanks, Wes.
So there's another question about the variability of Starry. So John is asking, how does year-to-year variability of Starry abundance and coverage compare with other natives like Kara? Would you expect there to be little change among natives year to year?
So I'm trying to think if we actually have data to get at this question. And we do have some data from those same biomass samples. We often measure the abundance of native species as well. And I would say that it's probably similar. There's years that are better and worse for all plants. But I think that starry stonewort goes through much more extremes because it has this potential to get these super, super high abundances, and it has this ability to do, you know, major growth. I think that a good year for starry stonewort, it's just gonna like, you know, it's gonna be more good, if that makes any sense. And so I think that the ranges are gonna be more dramatic for starry stonewort. I also think that, I don't know this for a fact, but my instinct would be that, you know, there might be different climate conditions. You know, if there was no invasive species, what conditions, you know, would be a really good year for Kara or Podmageton, you know, might be, I don't know if this is true, but I'm saying, like, you know, maybe they tend to be, you know, really benefited by early, like, you know, warmer earlier, and a really good year for starry stonewort is actually just a really long growing season into November or something. I don't know if any of that's true, but I could imagine that the climate conditions that are beneficial for those two different groups is not perfectly aligned. But absolutely, you would expect, you know, there's higher and lower abundance years for native plants as well. I'm sure anybody who lives on the lake could say, oh yeah, this year was, back in 1973, it was really bad or something like that. But again, I'd say the invaders, because of their ability to get such massive abundances, probably go through more extremes.
Okay, so we have a few questions coming in. I'll try to go through them in chronological order. So this one from Kat is asking, did you also look at water quality and how it might have impacted starry stonewort? So phosphate, nitrogen levels, etc.
Yeah, we haven't. That's absolutely in our work plan, and especially in some sort of additional projects that we want to do. But especially this year, we had just such limited ability to do field work that we weren't able to collect any of those sorts of samples. We were just going as quickly as we could. But that absolutely is our hope and our plan to pull actual samples and think about that in the future. The other thing that, the other strategy that we want to employ is a lot of these lakes, and in particular, a lot of these lakes have other sort of water quality, nutrient monitoring projects. We actually picked a lot of our lakes because they had sort of ongoing, again, often sort of citizen science campaigns that do this monitoring. So there should be sort of kind of lake-wide averages, more or less, for a lot of these lakes. And that's much like that climate data that I showed you those correlations for. We want to try and incorporate this nutrient data as well. We just haven't had a chance to get to that yet.
So these next two questions are related, so I'll go ahead and ask them together. So someone's asking, is there a native that can effectively compete with Starry? And someone else is asking, from a plant community diversity perspective, are there types of communities that show greater resilience for the expansion of Starry?
Well, I think that there are certainly, there are two-ish plant groups that I think tend to be it's hard to know exactly what the term strongest competitor means. The ones that seem to utilize the same types of habitats and that I tend to see as kind of right in there and also being sort of major space dominators. One are the native Karas, and then two, the Nitellas. The Naha, sorry, bushy pondweed or southern. or bushy, the naiads, I'm sorry, bushy naiad, southern naiad. And so those, I think, tend to be the ones that are most competitive and that might sort of displace or trade off with starry stonewort. But I think, the reality is that starry stonewort in a lot of cases is the stronger competitor. That's why it's acting as an invasive species. But I think that there are some interesting factors. Again, this is in our plan for the next couple of years to try and quantify this, but I think that probably a combination of sediment conditions and sediment and nutrient conditions, maybe sediment nutrient conditions, probably influence who is the more competitive species. My impression, though the hope is to actually get data on this, is that in sort of more sandier, rockier, less organic soils, the Caras tend to be a little bit more competitive. And so when the soil is a bit more organic, you might have starry stonewort, but then as it grades to sandier, rockier, the starry stonewort kind of peters out and the Cara sort of is able to continue maintaining that space. And then kind of on the other end of that, in sort of super highly organic areas is where we actually tend to see more of the naiads. But there I'm a little bit less certain as to whether, there's conditions that like the naiads are going to outcompete or not. The other thing that we'd see a lot, and this is kind of an interesting dynamic that I don't 100% know what to think about, but there's a number of sort of lily species that sort of actually are floating at the top. And I'm really interested in if you have these really dense lily communities at the top, can that sort of shade out the starry stonewort underneath it? Or is it one of those situations that like, they shade them out, but as soon as like an opening appears, starry stonewort can establish, and then it starts to displace the lilies. I don't know exactly what's happening there, but that sort of competition is another interesting place to look. But yeah, for me, I think it's the naiads and the Caras that have the best shot at being the most competitive, but on average, are they going to win or not? Probably. I think starry stonewort tends to win the competition, but not always.
So this next question is related to sort of climate change here. So they're saying spring months might be warmer in lakes under climate changes in the future, but that might reflect a shift in phenology where thawing and warming is happening earlier. Do you have a sense of whether starry stonewort is able to track with this shift in phenology? Would starry stonewort maintain their advantage in this case? Maintain its advantage?
Yeah, so that that is totally a really interesting possibility. That does just everything, the same pattern happens, but just a little bit earlier. I think the potential challenge with that is, are there components of light issues and light limitation that are sort of independent of temperature issues? And so perhaps a species could handle the temperature, but it still needs, a certain amount of light. And until you get to a certain point in the year, it doesn't have enough light to grow. So I think it kind of remains to be seen how that competition is going to play itself out. But so, because we have this latitudinal gradient down into Indiana where, we can see some of that future stuff, I think that that's the place where we're going to be able to get at these questions. We unfortunately don't have that sort of seasonal phenology data for our southern locations yet because we weren't able to do that this year. But that is absolutely, that is the part of the question that I am most interested and motivated about to, for next year, if we're able to actually do our full field season. I think thinking about that sort of early season dynamics, and is it different in our climate futures, which is Indiana, versus our climate presence in Minnesota. And so hopefully next year we'll actually be able to get at that question. But I think it's absolutely an interesting one.
So if anybody else has questions, feel free to type them in, but I'd just like to ask you a question that I think people, based on what people have been asking, I think they might be interested to know. So how long do you plan to continue this work? And it seems like a lot of the questions that are most interesting are sort of these longitudinal questions about changes over time to sort of get at this heterogeneity that you're seeing in the data.
Yeah, well, I'll say I think it was honestly a bit of a surprise even to me to see that there was this difference in 2020 versus previous years. I thought that mostly we would be seeing differences north to south, and that was going to be our avenue into thinking about these climate questions. But this actual year-to-year heterogeneity was surprising, interesting, and I think really quite important, which doesn't really answer the question. But so I think it's making, the idea, like if it had just been this north to south thing, I think the project could have sort of run on a slightly shorter time scale because we could have made that comparison a little bit more easily, just, utilizing the same year. But now I think that this year-to-year heterogeneity is sort of important enough that hopefully we'll be able to run the experiment a bit longer. We should, again, provided COVID, or provided we're able to work either through COVID or after COVID, next season we should be able to get out and do this again. And so we'll definitely have one more year of our full data set. And then I'm in the process of trying to acquire some additional funding to try and keep sampling for maybe another 3 or so years. But that is up to the National Science Foundation at the moment. So our plan is certainly another year. Our hope is another 4 to 5 years. And then we'll have to see if it's still useful to keep monitoring these or if we should maybe set up new locations. But that's sort of the longer term and less certain at the moment range part of the question.
There's one final quick question here. Based on your findings, what's been the max depth Starry stonewort's been found at?
I think it's about 12 to 15 feet is where I would sort of put it. And it's actually often pretty interesting. You'll get down there and it often is it's like a really sharp boundary. Like it doesn't sort of peter off. It seems to, there's often a pretty sharp line. Although I don't think it's purely a depth question because again, like turbidity, so in certain lakes, it'll probably be a little deeper and a little bit shallower, but in the lakes where we've seen it the deepest, I'd say in that 12 to 15 feet range is kind of the extent.
Well, thank you very much, everybody, for attending. We really appreciate it. I don't know if Renja wants to say anything else, but I know there's another session starting at 1. So if there's nothing, nobody has any final wrap-up thoughts, everybody can feel free to have a break before your next session.
I'll just say thank you all for your time. And if you have any other questions, feel free to send me an email. I'm on the MACERC website and at Indiana University's Environmental Resilience Institute. Thanks for your time. It's been really interesting, and enjoy the rest of your sessions.